Welcome you 12 to another video for our flipped classroom. Okay, so today we have a relatively long and large video. There's a bit of information to process. It's to help prepare you for conducting an experiment, or 3 experiments, into factors that affect enzyme activity. So let's just check out the dot point and go from there. So it's 9.2.1.S1. Identify data sources, plan, choose equipment or resources, and perform a first-hand investigation to test the effect of increased temperature, change in pH, and change in substrate concentration on the activity of named enzymes. So the S stands for skill building. Well, not really. That's the way I like to think about it. It's something that you are going to develop. It's not just handed to you on a silver platter. So sometimes we think of skills like this. He's so proud of himself. Oh my goodness. Oh my goodness, that just makes him so much more employable. Right? Wrong. So, we want to exercise your brain. We want to build this little bad boy up so that you become more employable. You become better to work with, better at problem solving yourself. And overall, give that little muscle a workout of yours, Go to the brain. Alright, unpacking the dot point. Let's check it out. So, our verbs here, we need to identify. So that's basically just highlight what we need in order to perform. Alright, so you actually have got to do something here. A first-hand investigation, which is the experiment, to test the effect of temperature, change in pH, and change in substrate concentrations on a named. This word named actually is kind of important to see this. Anytime you come across this, anywhere it says the word named, you actually have to have, it sounds silly, but you can't just kind of say like, you might say it says named ectotherm or something like that in Australia, and you can't just say, oh, snake. Like you actually got to be like red snake or something, or brown snake, or whatever. But named, you got to have a name for it. It's important. You can't just say an enzyme. Alright, so the one that we are going to look at is rennin. So, rennin is found in the gastric juices of mammals. And what rennin does is it gets the milk proteins that you see in milk, and it curdles them. It clumps them together. And you get milky solids that we can use in cheese. It's rather simple to make things like ricotta cheese at home. And then you get this absolutely disgusting watery filth called whey. And you might have heard of curds and whey from the Little Miss Muffet story. Alright, so it's quite hard to actually get rennin in a, it's quite hard to extract it or anything. So there's a commercialized version of it called RENNIN. And it's used in dessert tablets. These things called junket tablets. And that's going to be our source of rennin. So if you hear the word rennet, it's just a commercialized form of rennin. Alright, so before I launch into this slide, we should just be pretty clear about what we're trying to achieve here with this video. You're going to conduct 3 experiments, and we're going to give you a method, and we're going to kind of lay this experiment out for you so that you can understand the process that we're going through. And then by the end of it, you should have, you're probably going to have to watch this video a couple of times actually, you should have all the know-how and setup done so that when you go into the lab you can do your experiment, and then looking at your results easily make some statements about the factors that affect enzyme activity. Alright, so the first one is we're looking at enzyme activity and temperature. So what we're going to have is 8 test tubes set up in 4 different temperature water baths. Now a water bath could just be a beaker, one with icy water, one with cold water, one with warm water, and one with near boiling water. Now ideally you'd know the temperature of these things, but on the day we could try and say something like, I don't know, 85 degrees, but on the day we won't know that until we actually get the water there. So we're just going to give them these kind of ballpark figures. So icy water is going to be around 1 or 2 degrees, maybe in 3 or 4, I don't really mind. And then we're going to have cold water, Now, cold water could be somewhere in the range of 10 to 15. Then we're going to have warm water, and that's going to be 30, 35. I'm not too sure about that one. We'll see how it goes. And then near boiling, we really want to get it well and above away from body temperature. So we're looking at least somewhere over 65 degrees there. Alright, so what we're going to do is we're going to place 30 mils in each test tube. So we've got our substrate, which is the milk. We're going to place 30 mils of it in here. And then we are going to grab 5 mils of rennin, and we are going to add it to one of the test tubes. What this does is it allows us to kind of compare, well, does milk curdle by itself, or is it in fact the enzyme that curdles the milk? And so it's a kind of a control. Alright, in each of these test tubes we've got to keep the some variables the same in order to make this test valid. Validity is basically saying that you are controlling all variables, so it's only the effect of one thing that is determining the outcome of the experiment. And in that case it's going to be the temperature. So we need to keep the pH the same, and so that should be straightforward to do. We won't need to do anything with this. If they're all the same liquids and enzyme, they'll be the same there. We're going to carefully measure the amount of milk so that we don't have excess amount of substrate or less substrate in here. And we're going to carefully measure out the amount of rennin, and I'll show you a technique in the lab when we do that. Alright, so there's your experimental layout. Let's have a look at this. In each experiment, in order to be a valid experiment, we need to have easily identifiable variables. And so the first one in this one is the independent variable. So the independent variable is going to be the thing that we are able to determine. Alright, it's the thing that we want to test. So we have icy water, cold water, warm water, near boiling water. Right? We made the decision to make them those temperatures. Over here we have the dependent variable. Alright, so the dependent variable is the factor that we are going to measure. And so in order to measure something, you actually need an instrument to measure it with. So when you're thinking about these things, if you can't physically hold something in your hand to measure it, chances are it isn't the dependent variable. So we're going to have time, the time it takes for the milk to set. Alright, so the quicker the time, it means the higher the rennin activity, which means if it sets in 5 seconds compared in icy water compared to a minute in near boiling water, then we're going to clearly say rennin has higher activity in the icy water. Alright, quicker the time, higher the rennin activity. Alright, well, let's have a look. This is something that I'd like everyone to get down on a sheet of paper, a nice neat sheet of paper, before you start the experiment. So this has, we need to take measurements for both. Alright, so are we expecting the milk by itself to curdle in these temperatures? Probably not. Maybe. I don't think so. But we're having this just to make sure that it is only the enzyme that's affecting the ability to curdle. So this one is likely just to, I think it'll just stay the same. It'll just stay milky. But over here though, we are going to look at the effect of the enzyme on the substrate. And so we want to test the time it takes for the milk to set. So you've got your graph over here. And then we are going to actually do something to the data. We're going to manipulate it. Now, I want you to take, tilt your head sideways if you need to. It says time taken to set, and then in the brackets here it says 1 over x. So if you follow my mouse, if you're watching this one, right? So if we just had it time taken to set, it would suggest that, if we use that previous example where if it took only 5 seconds for the milk to set at freezing, so freezing's down this end of our scale for temperature, and so if it took only here, and then we go up to boiling water and it goes all the way up to here, we go, whoa, look at that, it's like increasing. That must mean that rennin is increasing, its activity is increasing. The graph doesn't kind of relay, it's not useful in helping to show the activity of rennin, because rennin down here is actually the fastest, which is the highest activity. So we flip it. We put 1 over x, and mathematically it inverts the graph that to really show the effect of rennin. I'll show you everyone how to do that if you need help. Come and talk to me or Miss Skelly, and we'll be able to walk you through that process. Not a problem.